Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is March 27th, 2025, and we're talking to Nace Golding. Nace is professor in the Department of Neuroscience and the Center for Learning and Memory at the University of Texas at Austin. His laboratory studies hearing and the functional arrangements of the neurons in the auditory pathway that's observed hearing. And a bunch of the auditory pathway, complicated, interesting, starts low in the brainstem, has a sequence of nuclei with unique functions as you ascend the nervous system, and then things become vague after that. There's a moment at which things become more vague. And that's places somewhere around the inferior colliculus. Maybe we'll talk about it today. Nace was here before. It was about 11 years ago. If you want to listen to his previous podcast, it's episode 111, February 13th, 2014. Of course, a lot has happened since then. So welcome, Nace.
Well, it's a pleasure to be back.
Also with us is Marina, UTSA's own expert on auditory midbrain circuits and frequent podcast participant. Hi, Marina.
Hi, happy to be here.
And I'm your host, Charlie Wilson. So Nace, the auditory system is wonderful and special because sound is so wonderful and special. And it's really great for neurophysiologists because neurophysiologists study activity of neurons in time, and sound is basically a waveform in time. So we can compare sound to neurons really well, compared to comparing sound to other things, or to comparing neurons to other things. So maybe that's why so much great neurophysiology has been done in the auditory system, or so much success has been had. Maybe you don't feel that way because you work in that way, never feel like success. But to me, it really looks like a lot. And the auditory, especially at the lower levels of the auditory system, there's a set of a sequence of nuclei that start in the brainstem and ascend up the nervous system. And the first few steps in there have pretty clear functions. And the functions have been identified with the function of individual cell types. So there's a cell type that does this to the sound, and a cell type that does that to the sound. And the nuclei are just homes of those cell types. We really identify function with cell types. That may not be completely right, but that's my perception of it. And so we need to understand cell types, I guess. So can you say something? Can you start by just telling us something about cell types and the value that's?
Yeah. In fact, my history, I think, is sort of rich in pursuing the question of cell types, because when I was a graduate student, I worked in this place called the cochlear nucleus, the first stop for auditory information in the brain, and it's such a beautiful system because it's like just this cast of unique characters. Every cell has its own colorful name, like bushy cells and octopus cells, and they have these kind of unique cell morphologies. They look different, they respond different, and it takes part in this process of immediately separating this homogeneous activity of sound being sent to the brain, and it separates it into different channels using these different properties, and really informally call it sort of these different personalities. You can think of each cell having its own electrophysiological fingerprint, and then
computational styles.
Yeah, that's right. And and what makes it a beautiful system is to sort of understand how ion channel types and distributions sort of factor into what kind of information they're computing. And so it's a great marriage of cellular and systems neuroscience in this area.
So how were those cells discovered? I mean, it was a while ago.
Yeah. I mean, it's because it's the first area of the brain. It was, of course, of the simplest to understand and of immediate interest, because the auditory nerve basically projects right to this nucleus. And so if there's any place that you're going to be able to figure out how the nervous system deals with sound. That's the first place to begin.
How were the cell types identified, though? It sounds from their names that they were morphologically defined.
Yeah, that actually sort of brings on some of the things we thought we'd talk about today, which is like, what is a cell type, and how do we, there's such a rich history in this. Just like in every brain area, it always boils down to the early anatomists who, with Golgi stains and other staining methods, just started looking at form and guessing as to what function these forms might be participating in. And for me, one of the influential papers that my graduate advisor, Donato Ortel, was very fond of was this paper by this Norwegian anatomist, Kirsten Ozen, who described the cell types in the cochlear nucleus using what criteria anatomists could use. And they were often what we would consider now to be kind of arbitrary criteria: how big the nucleus was, whether they had certain kinds of granules in them, some things that we would say are hardly functionally relevant now, but yet they were ways that they had those were the tools at the time to separate different, what they thought might be functional cohorts of cells.
And it worked.
Sometimes they were, and sometimes they weren't, actually. And I think some of those criteria were relevant, and others proved to be maybe variations on a theme within a cell type. But the cochlear nucleus was a fortunate place for a lot of these anatomists to start because there are such, I think, pretty radical distinctions between the different kinds of participating cells. But as you mentioned, it gets murkier as you go further along in the pathways, and maybe that's true for a lot of different brain areas and sensory modalities. It's easiest at the periphery.
Yeah. But it's sort of what's the right word for it? It's our paradigm, or maybe just an article of faith. We identify the cell types, we figure out what their functional properties are. We're going to look at those functional properties and go, oh, I see what this neuron's job is. And it's going to make sense to us. And it has worked that way a little bit, but not perfectly, I guess.
Well, one of the challenges for me when I was a student and just trying to learn the auditory system is that depending on who you were, everyone had their own terminology. So how we talk about cell types often depends on who we are experimentally. So the anatomists had their names: bushy cells, octopus cells, and then the engineering-oriented in vivo physiologists had their own separate terminology that was very engineering, onset units, choppers, named after circuit, electronic circuit behaviors, and other pausers, and they had their own separate terminology that mapped onto these anatomical cell types. So then you had cellular electrophysiologists who often had a separate terminology. So as a student trying to navigate this literature, you had to try to draw lines between these different labels to say, oh, this, onset unit means this type of anatomical cell type, or possibly this other one sometimes. And so there was this whole mapping that you had to perform as a student to try to understand who, just to get on top of the terminology.
It has finally become disambiguated, I suppose.
It's gotten better, but I think that persists.
That persists, I would agree.
And I think one thing that actually inhibits, I think, sometimes students from accessing what is interesting and exciting about some of the auditory research. Also, too, that there are so many nuclei, unlike, say, vision, where there's really a pretty limited number of stops on its way to cortex. There's more of a parcellation of duties in the auditory system amongst sort of specialized nuclei that often have less than memorable acronyms, because it borrows names from anatomists who described places like a trapezoid body, just sort of this trapezoidal shaped area, and then there were nuclei of the trapezoid body, and so you had great tongue twisters like the medial nucleus of the trapezoid body for one particular nucleus, which houses actually one of the famous synapses in the nervous system, the calyx of Held. So
Which is
just
its own interesting kind of name, also anatomical name.
yeah, it's named after the German anatomist Held, who described a lot of different structures in the brain.
So what's the current status of the procedure for identifying cell types functionally and anatomically, and I guess biochemically at this point we would want to be able to?
That's right. I think if anything, we're adding more layers of terminology onto these sort of historical definitions, because now we have molecular genetic definitions. So now, in addition to these other names, you have to know whether it's a calbindin expressing or parvalbumin expressing neuron, and how that maps onto the physiological and other anatomical definitions. And the hope is that the more criteria we keep adding, suddenly we're going to come up with a more precise definition of a functional cell type. But in some ways, I think we're going the wrong direction, and we're just making things more complicated without necessarily coming to a better understanding of how this all maps onto function.
Yeah, the hope was, I guess, must have been, when this started, that all these different ways of defining cell types would eventually converge to one set of cell types. It would turn out there's a one-to-one mapping between them all. But and to some extent that's true, but it's not completely true. There might be a physiologically defined cell type that contains 5 different markers for other cell types.
Well, I think things would be a lot simpler if, as an organism, we always sat in a quiet room under perfect listening conditions. But the fact is we're in a complicated environment, and as Marina studies, she's very interested in neuromodulation, and so depending on the context, all these definitions we've created for cell types often change depending on the situation that we're encountering. If we're in a noisy environment or we're under stress, so sort of the behavioral context can change how neurons respond. And so, as careful as we are about coming up with definitions of neurons, they're quite labile, I think, particularly now that the whole field of neuroscience is moving toward more realistic, instead of working on purely anesthetized, quiet animals with very simple stimuli, scientists are now able to study animals in more natural contexts where maybe some of these clear lines between cell types get even further blurred because of the action of neuromodulators and other factors that can modify how cells respond, and how they, maybe even the temporal and patterns in which they respond to different stimuli.
This always made anatomists not confident about physiologists' cell type definitions, because they thought, well, a bushy cell's always going to stay bushy, but chopper might turn into a posser. Right?
Yeah, that's right.
Yeah. And then, so there are a couple of changes that might make anatomists less, if there are any anatomists left, might make them less confident about their view. And one of them is anatomically things change too. Cells actually do grow dendrites and change their shapes.
Yeah.
And then we have all this transcriptomics, and that's super labile. And it turns out that transcription is constantly changing as well.
And even then, depending on how far you want to dig, someone would say, well, it's not all about the mRNA. It doesn't necessarily guarantee it's going to be expressed. Yeah. So translation and post-transcriptional regulation is also equally important. It can also modify the functions. So I think we're never going to reach the end.
Yeah, so how far can you go, right, to find a cell type?
Yeah.
You can get it to an endless division.
Should we give up?
Types.
I mean, are cell types still the only hope?
I think it's important to know your cell types, at least like in a functional way, right? Are these cells like they have like a similar function, like in the cochlear nucleus, there's a group of cells that have the same response to sound, for example. Where they're projecting to, we know their projection patterns.
My
So I
own
think
prejudice is that cell types is the only game in town. I mean, in the part of the brain that I work in, basically it's the keys to the kingdom.
yeah, we need
And
to.
cell types do keep their identity pretty well. And there is a commonality of function among
Right.
members of the cell type. I think as experimentalists, every time we see some little deviation, we freak out.
Yeah, which is not the right way to do it.
That cell types are not meaningful. But there are individual differences among cells of a type. And maybe we should embrace that and view that as part of what the cell type's job is.
Well, there's been a whole series of papers that I quite admired that really embrace this idea of sort of elastic definitions of cell types. Cell types aren't supposed to do exactly the same thing.
Yeah.
In fact, a lot of computational papers now profess that there's more information if within a cell type definition that each cell does not respond exactly the same way, is less stereotyped than their neighbors, because you can extract more information. There's more bytes of information that are being conveyed as a population when each cell has a slightly different sensitivity or perhaps receptive field or whatever measure that, depending on what circuit that you're studying. So there's room, I think, for elastic definitions of cell types, and maybe, as you said, we tend to freak out when things don't fit into a neat box, but maybe it's okay to not sweat the details too much.
Yeah.
So in a place like the inferior colliculus, where the search for cell types is going on,
it's going on
intensely, what are we have great new tools that weren't available when people were studying the cochlear nucleus in the beginning. What's our strategy for identifying the inferior colliculus cell types? And why don't we already know all the names of all the cell types?
Right. So the inferior colliculus, like for a long time in the field, it has been really difficult to identify those cell types. And that's because we have like cells with different morphology in the inferior colliculus, so broadly classified in these two groups that are the stellate neurons versus the disc-shaped neurons. But there's a lot of heterogeneity with the cells that have the same morphology. So they have different firing patterns. They can be either GABAergic or glutamatergic. So they produce different neurotransmitters. So the morphology was not enough in the inferior colliculus from the early days to be able to subdivide even between inhibitory and excitatory neurons.
And we really rely on that. I mean, if I told you, well, cells within a type are diverse, some are excitatory, some are inhibitory, my heart rebels at that.
Yeah.
Excitatory neurons ought to be a different cell type than inhibitory neurons. Right. So you have a bunch of cells that look alike, but some make GABA and release this. And
glutamate. And they also have, when you think like in the physiology way, they have similar firing patterns, so we cannot even classify them physiologically. We cannot classify them based on the way they respond to sound, right? They have very heterogeneous response to sound in the inferior colliculus as well. So as we move on in the field to identify cell types, we start to take advantage of molecular markers like neuropeptide Y, CCK, VIP, and classifying those cells as classes or cell types that commonly express those molecular markers.
So all the markers you just mentioned are neuropeptides.
Yeah,
are those the markers of choice? I mean, earlier I think you mentioned calcium binding proteins.
CCK, yeah.
Are there what's the range of different kinds of molecular markers? I mean, in principle, we could be looking at transcription factors.
Right. Right. So as for now, for the field, we have been looking into neuropeptides to identify the cell types. And that's because we haven't even done a transcriptional analysis in inferior colliculus, so an experiment like RNA-seq to be able to identify all those different gene expresses in the inferior colliculus. So this is ongoing in other labs that are looking into that. So hopefully we can have like a better idea of different markers in the inferior colliculus that people can go on and validate and see whether they have like same morphology or similar firing patterns.
So we sometimes joke, I know I have sometimes joked that, in fact, I think it was with you at one point when we were talking about, oh, you study somatostatin, you must be neurons, you must be interested in somatostatin. And the answer is, oh no, they're only just a group of GABA neurons to me.
There's also that.
But every time we hear one of those neuropeptides, it probably has some circuit function as well. So maybe neuropeptides are a great marker to use as opposed to just some gene whose expression is high in that neuron that we don't know what it does.
Yeah.
So, and you have been using CCK, so you use neuropeptide Y, you use CCK to identify two different neuron types in the inferior colliculus. Isn't that great? I have you both.
Yeah.
So what are the two neurons? What's the neuropeptide Y neuron? What can you tell us about that?
So those are GABAergic neurons, so they co-express GABA and the NPY. And they are stellate, a stellate population of neurons, so they, most of the time, like 95% of them have a stellate morphology. When we look to their physiology, they tend to fire sustainably. When we do a current injection and look to their firing pattern, we have some work in progress to see how these neurons respond to sound. Like what is if we play a sound, how is going to be the response of those NPY neurons? So
usually my way of dividing neurons always starts with, it's something I learned from college. Yeah. It's a long axon neuron, it's a short axon
They can be either, right? So we know that NPY neurons project locally in the inferior colliculus. They also send long-range inhibitory projections to the auditory thalamus and to the other side of the inferior colliculus, the contralateral IC. One question that we don't know is are those the same neurons? The same NPY neurons has axon collateral locally into the auditory thalamus, or there are different populations of neurons that are projecting to different locations.
So it could be long axon neurons that just happen to have local collateral.
Axon collaterals, yeah. Or it could be different populations that are projecting only local, but we don't know that.
When we were talking about cell types, I think that's actually one of the big criteria for a functional cell type is not just what shape they are or what they express, but where they go and who they're contacting. And that ultimately probably will be one of the big criteria for trying to really classify neurons in this midbrain area where it's so complicated. And there may be overlapping criteria for a lot of peptides and other things that neurons express, but ultimately they may have very specific projection patterns that might help to further classify what role they're participating in.
So in a lot of places that have long-range projections, there's been debates about whether there's a unique cell type that goes to each target or whether cells of a type project to many targets. The thing that comes to mind most is the cerebral cortex in the 1970s. That Jones claimed that for every cortical target there was a unique cortical cell type with different laminar distribution. And then it sort of, that seemed like based on solid evidence, but then once people got better methods, they found out that most of those cells were collateralizing a lot. And so the number of cell types that have unique targets from the cortex, so there's still some that exist, but there are many fewer. So those methods that sorted that out in the cortex are available everywhere for everybody now. So is the tendency toward discovering more collateralization or less in the stuff that we know about? So in your work, it seems like you have discovered a unique cell type that has a single target in the inferior colliculus.
Well, it's like 60% of the cells, so it's a huge proportion of the population, but it really only has one target. Everything that listeners of this podcast, every everything that they're hearing is going through this pathway, this major lemniscal pathway that is the main conveyor of auditory information up to higher centers. But I think ultimately, probably this class of cells probably could be subdivided, and probably will be subdivided into subdivisions of that might have, once we have, I think, more sophisticated methods of separating cells in terms of their targets and functionality, we'll have better ways of understanding how these populations are participating in different kinds of auditory codes. Right now, our methods are relatively crude.
But that neuron is sort of a backbone of the projections. I mean, normally in the little diagrams that we see, things make it into the inferior colliculus, and then from there they go to the thalamus.
That's right.
And there's some other connections, here
and
that
there.
mentioned in passing. But it can be confusing if those other connections are coming from the same neurons that are the main output, or whether they're coming from separate ones. So that one that projects to the thalamus has cousins all over the place, like the retinal ganglion cells that go to the thalamus.
That's right.
And they make very similar kinds of organizations. They have very similar way of connecting to the thalamus. The
connections across sensory modalities and the thalamic drive and the way that it's organized, and every sense has the lemniscal pathway and a next more modulatory pathway. So there you see the same themes repeated.
This is one of the things that seemed very reassuring to me when I was just beginning to study neuroanatomy, is I thought, look at the thalamus. There are all these really diverse things, inputs from the cerebellum, inputs from the visual system and from the auditory system and somatosensory system. And they all, when they get into the thalamus, they have this very stereotyped way of making synapses, these unique, identifiable, morphological entities that do the synapse, and they have the same kind of convergence and divergence patterns, and they produce the same kind of responses in thalamic neurons. Isn't that great? It means that we don't have to figure out every single pathway and every single neuron in the brain. Can I still think that?
Well, there's this new, we think this is a high level of detail, but with the emerging field of connectomics and these enormous data sets that are being generated with, EM tomography, and pretty soon we're going to have detailed wiring diagrams of every, essentially every cell in a nucleus, understand who they're talking to, what the connections are, the nature of the synapses at the level of EM. And that is, I think, rather than creating unifying or simpler categorizations, things could get just even more complex, because we'll have finer and finer details that are going to become available to us through these new technologies.
Yeah, when we get those, one of the problems with getting all of this detail is that we don't have any kind of context in which to interpret some of the detail. So it's hard to know which connections are the most important when we're, the old way of studying the nervous system was people would ignore a lot of things they saw. So an anatomist would inject some tracer into some part of the brain, and they'd see a main connection that went someplace, and then others that went other places. And they'd go, oh, those others, maybe those were just kind of artifacts. This is the main thing. They'd report the main thing, and and that sometimes turned out to be wrong. The main thing wasn't main at all, and the other one was. But there's some judgment was being used all the time to keep the complexity under control. And do we have a method, let's say we have these high resolution connectomics, like what they have for Drosophila. Say we had that for the auditory system right now. Are you frightened that would just mess us up worse?
Well, when I was a graduate student, I was really fond of the retinal circuitry because it was such a beautiful circuit, and you had these discrete cell types, and there were a limited number of them. And at the time when I was beginning graduate school in 1990, that was a circuit that was tractable.
Yeah.
And it was peripheral, so you could get at it, and the methods were there. And now with the explosion of molecular genetic tools, I don't understand the circuitry anymore, because for every bipolar cell, there's now a whole plethora of subtypes and subdivisions of every cell type based on molecular markers and expression of different proteins and detailed connectomic maps of who they're talking to. I don't understand the retina. I thought I understood the retina reasonably well when I was earlier in my career, and I've completely lost track of it now because the explosion of information has really overwhelmed, I think, the average understanding of. It's become sort of the realm of experts. I think that's the challenge. I think as each field, the retina is tractable, so I think it's a little ahead of some of the other sensory modalities or brain areas. But as this kind of detail starts to get heaped upon us, it's actually we're going to encounter the same problems. How do we sort of manage all this information and still make it tractable to understand, sort of cut through the detail and still understand sort of global processes?
I think maybe scientists have two jobs. One of them is to collect the facts, and the other one is to make sense out of them. And right now, there's no premium on the second one. We basically are just being encouraged to collect incredible amounts of data because that's what we get rewarded for. And of course, you'd like to make sense out of it as well as you can, given the time required while you're putting this piece of work together and writing it for publication. But that's not the number one thing. The number one thing is the data itself.
Yeah, but that may be the function of one of the uses of AI is it's really good at managing enormous, plowing through enormous data sets and finding patterns and
Yeah. We
don't have to categorize
things. We could
things.
just be the technicians for the AI.
Yeah.
And they will collect their data.
I mean, to some extent that's funny, but it's also quite probably true. Right?
Yeah.
Right. We're gonna hand over the heavy computing to artificial intelligence and then let it tell us what it finds, because we're gonna lose track of. It's just too much data.
And we've never been really good at interpreting our data anyway. My PhD advisor once told me that all results are true, all conclusions are false, and encouraged me to not read the discussion sections too closely. The author is just trying to pull the wool over my eyes. Read the results and make up my own mind.
Well, there's some truth to that, or some wisdom to that, but maybe taken to maybe not to that complete extent, but
he was exaggerating.
Yeah.
So where do we stand in inferior colliculus? How many cell types have been clearly identified? I've heard of basically 2 in here today.
In addition to NPY and CSK, we have VIPs and somatostatin.
Oh, so there's 2 more.
Somatostatin, yeah.
And so the CCK neuron is like 60% of all the neurons. So now we're dividing up the other 40%. So how many are there?
So VIPs are only 3% of glutamatergic neurons. NPYs are anywhere between 40 to 50% of the GABAergic neurons.
And GABAergic neurons are what percentage of all neurons?
25 to 35% of all neurons in inferior colliculus.
So there's still a missing 10 or 15% of
We are missing.
neurons that are not is CCK.
There's a lot of work to do in inferior colliculus, as
There
we
is.
were talking earlier. Yeah.
I'm just trying to get a rough back of the envelope idea about how many.
Yeah,
what
and the inferior
proportion of
colliculus has a lot of neurons, so it's estimated like five more times than all the auditory brainstem combined.
Oh, isn't that interesting?
I know. So there's
Why
a
does
lot
it
of
need so
neurons
many
there.
neurons?
Yeah, I know.
It's also one of the most metabolically active
Good
areas in the brain,
brain.
if not the
Yeah,
most metabolically
Yeah.
active.
Regenerative, right?
Speaking of early anatomists, they started to do metabolic stains, like the inferior colliculus is the one structure that lights up like a Christmas tree.
Yeah.
So it's
What's that about? Is that
firing
auditory?
all the time.
Yeah.
It's just incredibly highly active.
So does it need input to fire, or would it
No.
just go on like that?
Yeah. It fires at fairly high rates all the time, then can increase that rate in response to sounds or decrease them depending on the context.
It seems like neurons that respond to sound ought to be fast firing to begin with because they need to encode little time differences that are important for sound. Is that not?
By the time it gets up to the inferior colliculus, those codes have already been converted into some kind of a rate code. And it's a bit of a mystery how that gets folded into the circuitry that's conveying other types of auditory information. We use spatial cues to sort of separate out voices and things from their surroundings that make for a complicated environment, and somehow it's mixing with other pathways, but we don't have a good grasp right now of how that happens. And because we don't know the cell types, it's sort of a stumbling block because until we know how to define these other circuits, it's really hard to assemble a framework with which to interpret how these different pathways mix, because we start to get into the conundrum of what actually is a pathway, or what's a meaningful division of neurons.
And the inferior colliculus, is that the only way into the auditory thalamus? So without an inferior colliculus, the cortex never gets anything from the thalamus? Is that true?
We used to think that's true, but actually just in recent meeting I visited, there's been some pathways that actually circumvent the midbrain entirely, jump from the first stage of integration in the brain, the cochlear nucleus, going all the way to the thalamus.
Yeah.
To some special thalamic nuclei.
It's the express route instead of the local.
So, but surely people know what would happen if I didn't have an inferior colliculus. What would happen to my hearing? I mean, would I tell you that I don't hear, even though I might be able to hear something?
A lot of the thought of losing your inferior colliculus or not having your inferior colliculus would compromise your ability to understand the speech. This is one of the thoughts of.
So I could still hear.
You can still hear, right? But you cannot make sense of sound.
So speech is the obvious thing that I would have to make sense of. Could I appreciate music?
No, that's interesting. I was just thinking about this topic because, now the first neural machine interface was the cochlear prosthesis. Mm-hmm. And it's actually a really well-known phenomenon that it's actually quite amazing that people who have even a single prosthesis can understand kind of crude speech patterns. And with the aid of lip reading, they can follow conversations quite well. But music is actually something that's very difficult to encode. Music sounds terrible to people. People who become deaf, who previously enjoyed hearing, lose their enjoyment of music because the cochlear prostheses cannot encode the kinds of features that we find enjoyable in music and other.
Do we know what those features are? I mean, do auditory physiologists study music appreciation by playing musical things that sound good and musical things that don't, and asking how the neurons respond to them?
I think in animal models that would be probably more.
The topic always been interested in, particularly since, I have a history in the limbic system as well as the auditory system. And ultimately, these auditory pathways, it's probably the connections to the amygdala that give us a sense of emotional, the emotional valence of a piece of music.
And there are projections, right?
Whether you're into heavy metal or you're repulsed by it, either way, you have an emotional response to it, positive or negative.
We have projections from thalamus to the amygdala, right?
That's right.
Yeah,
it's probably one of the major
to the amygdala.
So I don't think the cerebral cortex is the place.
We forgot about cerebral cortex.
The cerebral cortex is the ultimate.
That's a projection as well. So there's really the two major pathways. There's a direct thalamic pathway, and there's also a cortical pathway, which is sort of a triangle because the thalamus activates the cortex and then comes back down to the amygdala. But there's something about just stimulating artificially these auditory pathways that somehow doesn't recapitulate the patterns that we find enjoyable in music. It's just too crude. So I think music is a subtle coding of information that is not easily done with the crude sort of stimulation techniques that are currently used for prosthetic devices.
Well, that's a goal. When everything is known about the connectomics, I will ask the AI to tell me where music is recognized and appreciated in the circuit. So anyway, thank you very much, Nace, for joining us. It's been fun talking about cell types. Thank you.
That was great. Thank you.
Thank you.
That was great.
It's been a pleasure.
This has been Neuroscientist Talk Shop.